In this syllabus, I will talk on the first unit, that is history of microbial. So in this presentation, I will talk about the all these aspects or sub points which are grouped in unit 1, that is history and development of microbiology, how different generation, how different lab generation theory or method, like that is spontaneous and non-spontaneous generation, classification of organism, and major characteristics of different prokaryotic eukaryotic cells. What are the techniques that are used in the identification of bacteria, and important group of bacterial group, that is prokaryotic cell, like photosynthetic bacteria, BGA, chematographic bacteria. Another important group is archaebacteria. So I will also discuss about the basic properties and the types of question that can be asked in ARS examination. Last year, virus, their properties, their classification, and their lifespan cycle. Beside some minor organisms like thyroid primes, I have also discussed these points in my presentation. So if we start from history of microbiology, actually I will focus on the historical development from the early era of 17th century, because there are some minor development before this era, but regarding ARS examination, question generally are not asked on this area. So those development which are mainly related to the development of microbiology as a discipline, so a question will be asked on that area. So I will start from Robert Hooke, because Robert Hooke was one more thing. Actually, I will not discuss basic property and somewhat in detail, but I will try to. Hello. So I will try to decide. I will try to discuss with you how questions from particular topic, particular subtopic can be asked in ARS examination. So it will be better for you that while preparing for ARS exam, you can read in that perspective. Okay. So you means don't think that I will cover all the things from A to Z in one presentation, because simple for us basic microbiology is somewhat different course. So that means this whole syllabus generally we cover in 2 to 3 courses. So it is very impossible to cover all this aspect in single lecture. So please note this point. So I will try to focus on the area which are important from ARS exam. So this is the Robert Hooke and his contribution. So he discovered cells, he pointed towards cells. He also discussed some microscopic fungi using microscope, and he has developed a microscope having magnification of 30x. His finding was published in Microbial. So regarding this scientist or this researcher, question will be asked for 2 marks, maybe 2 marks, or maybe for objective type question, means who coined the term cell. So what was the magnification of Robert Hooke of his first microscope, 30 magnification, like where his findings were published in which book. So such type of questions are being asked in the ARS examination, mostly for 2 numbers or objective type question. Okay. So in case of detailed information, you can get this information from internet or from any reference book. So it is a suggestion for you. In addition to this information, other related information you can read from reference book. Are you getting my point now? All of you. Hello. So is slides visible to you, all of you?
Yes, sir.
Okay. So this is a very important person. Actually, many of the times questions are asked on this scientist, because he is considered as father of microbiology, and he was first a microbiologist. So almost I think almost during every exam, ARS exam or SRF exam or IRA PhD entrance examination in microbiology discipline, questions are there on this scientist or this microbiologist. So you have to know the detailed contribution of this Anthony Levin group. His some of the contributions I have mentioned here, like he was father of microbiologist. Okay, uh, he has a hobby of making lens and microscope. Uh, he made a microscope using his own hand, and the magnification of their microscope was around 50 to 300 times. This is a major contribution. He was the first person who visualized bacteria and protozoa using micro, that is single cell organisms. He called them as animalcules. Okay, animalcules. This Antonio Lulu called this tiny organism as animalcules. Uh, and due to this finding, actually this led to the foundation of bacteriology and protozoology as a major discipline. Okay. And all of his findings are communicated and published in Royal Society of London. So, regarding the questions can be for two numbers or can be for five numbers. Five numbers. I think in year's examination, there are 20 questions for five number. Is it? I'm right. 20 questions are for five number, and 10 questions. Sorry, six questions are for 10 numbers. Okay. So questions on this methodologist or researcher will be for mostly for 5 minutes. Okay. So you have to keep in mind and read according in that perspective. So and one more thing, I am going along with the timely means time. The previous time with latest announcement. So how latest announcement has occurred in microbiology. So with that time duration, I am going to cover. So before first was Robert Hooke, his period was 1635 to 1700. Second was that Anthony Hooke, his period was 1632 to 1723. So during this period, actually, this is a very important area. During this period, there was concept of spontaneous generation as non-spontaneous generation. So this unit is this is the syllabus in our ERS syllabus. So you have to read this concept actually. So during 17th, 18th century, there was some research were proponent that living organisms rise spontaneously from non-living organism. So this theory was spontaneous generation, and some other researchers believe that living organisms arose from only living matter, not from decaying organic matter or not from other non-living matter. So this is a difference. This is a difference actually for theory of spontaneous and non-spontaneous generation. We call it as abagenesis versus biogenesis theory. Means, uh, abagenesis means spontaneous generation. Abiogenesis means non-spontaneous generation. So you have to keep the difference between what is the difference between abiogenesis and biogenesis, and who are the scientists that is proposing abiogenesis, and who are the researchers or scientists that were proposing biogenesis. So this is the difference I have mentioned here. You might have read actually this difference. Abiogenesis means life occurs from non-living organisms or decaying matter. Biogenesis means this hypothesis say that living organism give rise to the living organism. So there are some researchers who supported the abiogenesis theory, and here are some researchers list of researchers who supported the abiogenesis theory. So in case of errors examination, the question will be asked for prime number, differentiate between abiogenesis and biogenesis theory. So you have to keep these points in your mind. And one more thing, lastly, I will mail this presentation to all of you. So you don't need to write this as this point. Uh, it will be sent to all of you. Okay. So in theory of spontaneous and non-spontaneous generation, mostly questions are asked the one uh, who are the researcher who disproved the theory of spontaneous generation. Spontaneous generation means who disproved the theory that living organisms are generating from non-living organisms. So there were some scientists who supported spontaneous generation. And regarding the air cell generation, these are some four scientists who disprove the theory of spontaneous generation. They give the theory of biogenesis, that is, particular living organisms occurs from free-living organisms. So, in some cases, question can be asked for 10 numbers. It means write down the proponent of spontaneous generation theory and describe their experiment. So you have to keep in mind that all the scientists who carry different experiment to disprove the theory of spontaneous generation, you have to keep in mind that their experiment trial. So in case of Louis Pasteur, that is Swan experiment. So related experiment of all the scientists you need to be keep in mind, because most of the times questions are asked on for 5 numbers or 10 numbers. So these are some pictorial graphs I have shown here. I will not go in detail regarding their experiment, because all these informations are available in basic reference book, or you can get information from internet. Slides are moving now.
Yes, sir.
And slides are visible to all of you.
Yes, sir. Yes, sir.
Okay. So, with advancement in period, I will discuss John Tyndall. John Tyndall was one of the scientists who disapproved the theory of smoking generation, and he was proponent of theory of germ theory of disease. Germ theory of disease means particular microbes is responsible for positive position of particular disease. Means that is germ. Germ means microbe or living tiny microorganism. So this is a theory given by Tyndall. Also, Tyndall discovered the endospore. Endospore is a types of resistance spore in bacteria. So he discovered this endospore. Actually, in addition, he has some contribution, like he discovered two types of form of bacteria, like heat stable form, that is endospore, which is resistant to heat and other stress factor, and other is heat sensitive form, that is physical. So there's two forms where there are presence of two forms. This was discovered by John Tyndall. So in order to kill the endospore, because endospore was resistant to heat, acidity, or high temperature. So in order to kill this endospore, he discovered the new process, that is the intermediate heating of liquid media or any solid media. So this process was called as Tyndallization. So many times questions are asked about two numbers. What is Tyndallization and who was founder of this Tyndallization? So you have, and what are the application of Tyndallization? So you need to keep in mind what is Tyndallization, its definitions, and its concept and its application. So you have to keep in mind most of the times question can be asked for two numbers, objectives, fill in the blanks, or for five numbers. Then another researcher is Louis Pasteur. He is a considered as most important founder of microbiology, because due to his various and diverse applications, diverse discovery, and his finding in the area of microbiology. Along with Tyndall, he was also one of the proponents who disapproved the theory of abiogenesis or spontaneous generation. He gave the principle of vaccination, microbial communication, and pasteurization. So we have to keep in mind. One of finding is he was one of the three main founder of bacteriology, along with Ferdinand Pohl and Robert Koch. He had proposed the principle of fermentation or preservation of food. He gave sterilization techniques and developed a steam sterilizer for a wound and antibiotic. As a researcher, he has given method of milk sterilization using pasteurization. He contributed to the vaccine development against anthrax, cholera disease, and rabies disease. He was one of the proponents who gave the theory, germ theory of disease. That means particular disease is caused by germ, that is tiny microbes. He gave the concept of liquid media. So there are some his contribution. One of the contribution is he has founded a founder of Pasteur Institute at Paris. So most of the question can be asked, who founded a Pasteur Institutes. So in that perspective, you have to read and keep in mind. Likewise, Rabokoj is he had established a relationship between Bacillus anthracis and anthrax. He had got Nobel Prize in 1905 for the discovery of tuberculosis bacteria. Due to his medical science discovery, he is considered as father of practical bacteriology or medical microbiology. Some other notable contributions of his are he has introduced the solid media for bacterial culture growth, that's methods for isolation of bacteria in pure culture, hanging drop method for visualizing whether particular organism is motile or non-motile, discover bacteria like anthrax, chlorella, and tuberculosis bacillus. Like another aspect is he gives staining technique by using annealing knives. And last one is major contribution is coach postulate. So this is a very important from exam point of view. Questions are asked. What is the question can be asked for 10 numbers? Give the contributions of Robert Koch and what are the Robert Koch postulate and explain it. Explain them. So these are Robert. This Koch postulates you might have read. Koch postulates deal with that particular organism is responsible for causing particular disease. So this is the experiment he explained actually. This is his experiment. So you have to keep in mind how core postulates are responsible for establishing a particular that particular organism, that is microbes, is responsible for causation of particular disease in human being or any other living organism. So you have to keep in mind contribution of this Robert Koch, and from exam perspective, you have to prepare maybe for 5 numbers of 10. So here I have shown golden age of microbiology. Uh, as far as I remember, during my time, they have asked a question for 10 numbers. 10 numbers means what is the golden age of microbiology? Actually, the period from 1857 to 1910, that is considered as golden age of microbiology because many of the new discovery notable contributions were made from different researchers, so that led to the development of microbiology as separate discipline. So in this regard, you have to keep in mind the major contribution that have occurred between the period from 1857 to 1910. Because question can be asked for 5 numbers or 10 numbers. What are the golden age of microbiology? And describe the notable contributions of microbiologists or researcher during the same period. So you have to keep in mind and prepare in that way. So notable contribution, as I discussed, like individual contribution, Linnaeus taxonomy theory, like pasteurization, Robert Wood postulate. In further slide, I will discuss about immunogeography, gram staining. That this beside this, there were some other researcher. You need to keep in mind who were the active researcher during this period and whatever their contribution during this period. So you have to prepare and write in answer. So, another scientist is Edward Jenner. His period was from 1749 to 1823. He is considered as father of immunology. Questions are asked for objective types. Who was the father of immunology? So we have to keep in mind this. Uh, he developed because he is called as father of immunology because he developed the first vaccine of world, that is smallpox vaccine, and he used a copy virus to immunize children against smallpox, from which the term vaccine was derived. So you have to keep this information in mind. Another notable researcher was Joseph Lister, and his period was from 1827 to 19. And he is considered as antiseptic surgery. He is considered as founder of antiseptic surgery because he used carbolic acid, for which we call as phenol, and for as antiseptics on the wound or during surgery operation. He instructed surgeon under to clean the gloves, wash their hand before and after operation with 5% carbolic acid solution. So due to this, his work and contribution, he is considered as father of antiseptic surgery. These are questions means on this area. Questions can be taught to members on objective type questions. Uh, Another is elite Metchenko. Uh, Actually, he has discovered the phenomenon of phagocytosis. Phagocytosis is a cellular concept of immunity. Phagocytosis means actually he did a lot of work on phagocytosis action at Pasteur Institute. He is one of the followers of Louis Pasteur, and he found that uh, in our cells, in our human blood cells, that leukocytes, they are phagocytosis. Phagocytosis means they are able to attack the invading bacteria and kill them. So this phenomenon is called as phagocytosis, and this was discovered by this particular scientist. Other is for English, he is considered as he gave the techniques of chemotherapy. Chemotherapy means uh, use of particular chemical as a, and it's used as a drug. So he produced the ARS phenomenon, an arsenic compound, the first synthetic drug for destroy of syphilis microbe in the human body. So this is a very important contribution you have to keep in mind because questions are often asked on this area. Later, he discovered a synthetic arsenic compound that is sulfur. Because many times questions are asked, these are mostly objective questions. The name of compounds are being are more often asked. What is the name of compounds which were called as magic bullet? This particular compound was effective against syphilis disease, and it was named as magic bullet. So you have to keep in mind this. If you have any questions, you can ask me. Now, till now, I discussed about the contribution of basics microbiologist. Here is a contribution of microbiologists who did pioneer work in the area of soil microbiology. So one of them is Martinus Bejan. He was famous soil microbiologist, and he had given a lot of novelty and discovery in the area of soil microbiology. First one was she gave the enrichment culture techniques. Enrichment culture techniques for identification of particular group of organisms. Along with Sergei Vinogradovsky, he was one other researcher. Both of them worked on the concept of enrichment culture techniques, and these culture techniques allow us to isolate the highly specialist organism from any natural habitat. Later, he cultivated and isolated Bacillus radicola, which was later named as Rhizobium leguminosum. The bacteria is responsible for nitrogen fixation in leguminous. He also characterized bacteria as free nitrogen fixing bacteria and isolated new bacteria Aerobacter. Again, he also worked on sulfur reducing bacteria, sulfur oxidizing bacteria. Besides soil microbiology, uh, by studying the tobacco mosaic disease, he concluded that uh, filterable pathogen was contagion during freedom. He turned a point to convey his concept of living infectious agent in a fluid, not cellular. That is viral. It is for viral infection. So this was some brief contribution. Besides this, some other points can be added here, but I have just given few points here. For every researcher, you can search some other if there is a remaining other contribution, and you can add to the this notes. Another was Sergei Vinogradovsky. I discussed in previous slide. He was also founder of enrichment culture techniques, and he did this techniques. He used this technique for the isolation of bigger species. Uh, he discovered and explained the chematotropic nature of bacteria, which I will discuss during my subsequent stage of presentation. Later, he identified the process of nitrification, isolated nitrified bacteria, azotobacter glucose, and showed that they are capable of nitrogen fixing. Due to this contribution, he is considered as father of soil microbiology. This particular scientist, he has a lot of contribution in soil microbiology. So because these two researches are very important in soil microbiology, so questions are more oftenly asked on this researcher. So we have to keep in mind, we have to prepare in that way because they are two very important persons. Uh, like cumulative nutrition of soil bacteria. We also discovered anaerobic nitrogen fixing bacteria, Clostridium fasciculum. So these are some contributions. There is nothing like concept you can read from any reference book, or you can get more information from internet. You can gather information from internet books. So you can prepare in that way. So these are some individual contributions, individual in detail contributions of some researcher. Uh, only I mentioned this contribution because they are some major founder and pioneer work in soil microbiology as well as basic microbiology. Most of the times in errors examination, 90% of the questions from the history of microbiology will be focused on among these researchers. So that's why I have given these researchers uh, so that you can remember and you can read it. So in addition, during the 20th century, in previous slides, I discussed during 17th, 18th, and 19th century. So in addition, there are some development during 20th century. We have to keep some notable contributions that are related to microbiology because questions may come from this area. Like for example, Alexander Fleming, so who discovered penicillin, uh, such type of questions like Stanley experiment on tuberculosis of fungus. So this type of questions you can be asked in aerosol emission. Like in 1941, George Biddle and Tatum, they got Nobel Prize for their one gene, one enzyme concept. So you have to, I think during last time they were asked a question for find number. What is one gene, one enzyme concept? Describe and describe it. So because although this is not related to microbiology, but it is related to living organism. So it can be related to the plant or it can be related to the microbes. So questions may come from this area. And question how can? So you have to prepare that way because question may come for objective type question, two number questions or hundred question. That ivory characteristic contribution of how DNA is a genetic material. So their experiment you need to read and prepare in exam product. That's an Crick model of the DNA as double helix structure. So we have to read because questions are came for members. So like that, some other contributions or other important contributions we have to keep in mind and read. So these were some historical development in the area of microbiology, in the area of soil microbiology. Here, I have tried to focus most of the notable scientists whose work have given the discipline of microbiology. So, in addition, there are some other researchers whose contribution I have not included in my presentation due to shortage of time. Or so you can read their contribution. So now we will move on another aspect. Uh, After history of microbial development, we will go for the classification of living organisms. So during the from 18th century, there were many authors who tried to classify the living organism on different bases. So first was Carl Linnaeus. During mid 18th century, he classified the living organism in two groups. That's plant and animal. So I will not go into detail. Though you should focus on latest two development like Whittaker 5 domain classification and Carl Lewis 3 domain classification, 3 domains and 6 domain classification. So because these are most accepted classification of organism and other classification are not accepted. So questions are not asked on the first four class type of classification. Most of the questions will be on Whittaker and Carlos classification. Regarding the questions in microbiology, I am talking about questions in microbiology. Questions may come on the area on the classification given by Whittaker and Carlos. In case of Pythagoras, he gave the classification of organism into five kingdoms. That is Monera, Protist. Monera means bacterial group. That's prokaryotic cell. Protist means protozoa and algae. That is eukaryotic cell. And other that is all are eukaryotic, fungi, plant, and animal. So you have to keep in mind the Pythagorean classification. And question may come for object type of questions. From this area, we are not expecting that question can come for final or something. So from objective point of view, you have to keep in mind the authors or researchers and their types of classification. So a little bit, I will discuss about the Pythagoras, Robert Pythagoras high kingdom classification. So based on the different parameters like cell type, cell organization, cell wall, nutrition class, mode of nutrition. So based on this parameter, it did divide organism into five different kingdoms. Like as I discussed in previous slide, that prokaryotic group was Monera, that included bacteria, and eukaryotic group was again in eukaryotic group he divided into two groups. That is one group with unicellular structure like protista, which have protozoa and algae. Other was multicellular group. Again, in multicellular group, those group with cell wall and those without cell wall. So without cell wall was Animalia kingdom, and with cell wall was Plantia and Fungi kingdom, with phototrophic and heterotrophic nature of nutrition. So you have to keep this in mind. This is a Carl Lewis three-domain basic classification. So in some latest period that's during 1977, he identified some differences in prokaryotic means among the bacteria and archaea, and he proposed a new system. So he have given three-domain basic classification. Three-domain means domain bacteria, domain archaea, and domain eukarya. So in domain bacteria, there is a kingdom bacteria. In domain archaea, kingdom is archaebacteria, and in domain eukarya, that is there were four major domain like kingdom protista, kingdom fungi, planta, and animalia. So this type of questions for two numbers you have to keep in mind. You have to keep in mind and remember this. Another one was Woos Fox classification. This is the latest classification. They have divided all kingdom in three superkingdom. That is Eubacteria, Archaebacteria, Eukarya. And their major classification is based on the rRNA because that is phylogenetic classification because this particular RNA molecule is hereditary molecule and this is conserved molecule. So based on this RNA sequencing, there is phylogeny. They have divided all six kingdom into three major kingdom. That is Eubacteria, Archaebacteria, Eukarya. So questions make up for two numbers. So this was our classification of this is a this is a historical representation showing the size of microbes related to the other organisms. So these microbial cells can be visualized during using light microscope. But if you have to go for surface structure or some cellular organelles, you have to go for electron microscope. So this is just the size of different cell organelles, cell microbes in relation to the other organelles. Like bacterial size is around 1 micron. In case of plant cell size is around 10 micron to 10 micron to 200 micrometer. So yeah, sometimes why I showed here. Sometimes for objective questions can be so what is the size of bacteria? What is the size of bacteria plant cell? So just for that, giving that information, I have shown here the relative size of bacterial cell, plant cells, along with other cellular structure. So after initial two section, we will go for the classification and measure features of different microbial. So as I discussed in previous classification, there are major six kingdoms. So major six kingdom includes bacteria, archaebacteria, fungi, protozoa, algae, and viruses. So these classifications are based on the morphological, phylogenetic, physiological, and nutritional characteristics. So based on this different parameter, these microbes have been grouped into different groups. So actually, this bacteria archaea, they are prokaryotic cell, and this red highlighted fungi, protozoa, algae are eukaryotic cell. You might have read this. This is there is nothing new in level. And this is a virus. So virus is a cell. So this is intermediate between living organism and non-living organism. So before going for their individual characteristics, I will show basically the difference between prokaryotic and eukaryotic cell because the prokaryotic cell includes bacteria and archaebacteria, while eukaryotic cell include uh, fungi, protozoa, algae. So we have to know the basic properties of this bacterial cell, this basic cell property. It means if you are knowing this properties of this prokaryotic and eukaryotic cell, then it will be easy for you to remember their characteristics. This is a major actually topic. Questions are asked for 10 numbers. Differentiation between prokaryotic and eukaryotic cell with suitable examples. So you have to keep in mind these differences point. Um, I have given here difference between prokaryotic and eukaryotic cell. Uh, the major difference is as I discussed the size of prokaryotic, that is bacteria, archaea is somewhat less as compared to the eukaryotic. Their cell division is different size. Cellular organism, bacteria and archaea are unicellular, while eukaryotes are mostly multicellular. Cell wall, we have to. This is one major difference. Uh, in cell wall, there is a presence of peptidoglycan, protein and lipid, while these compounds are absent in eukaryotic cell. And in case of eukaryotic, the cell wall is mostly made from cellulose and chitin. Plasma membranes, that is cell membranes, they are present in both type of cell. Where one more difference is there is a presence of there is absence of sterile in prokaryotic, and there is no sterile in eukaryotic. Sorry, I was wrong actually. There is no sterile in prokaryotic, while there is a presence of sterile in eukaryotic cell. Okay. Metabolism mostly prokaryotic metabolize compound either through aerobic growth or anaerobic growth, while eukaryotics they are purely aerobic metabolism. They have purely aerobic metabolism. DNA is circular in case of prokaryotic, and in case of eukaryotic cell, it is very long linear DNA bounded by nucleus membrane. Uh, in case of bacterial cell, that's prokaryotic cell, we have presence of plasmid, while it is absent in eukaryotic cell. Like histone, here it is absent, they are present in eukaryotic cell. Synthesis, so there is also difference in relation to synthesis of RNA and protein, membrane organelles. Ribosome is here is 70s type ribosome. In case of eukaryotic, it is a 80s type ribosome. So lysosomes are absent here. It is present in eukaryotic cell. Locomotion. In case of locomotion, there is a difference between locomotion. Flagella is present, but their structure is different. So you have to keep in mind. Uh, Sometimes questions can come. What is the difference between flagella of prokaryotic cell and eukaryotic cell? So you have to keep in mind what is flagella, its function, and its comparison with eukaryotic cell. Galactocalus is a extracellular slimy polysaccharide containing material around the cell wall of prokaryotic. It is present in prokaryotic cell, while such types of structures are mostly absent in eukaryotic cell. One more thing is cellular respiration. That is aerobic or anaerobic respiration in bacteria, that is prokaryotic cell, that occurs mostly in cell membrane. But in case of in case of plant cell, that is our eukaryotic cell, it occurs in mitochondria. So you have to keep in mind. Their examples are bacteria, archaea, and in case of eukaryotic cell, that examples are fungi, algae, and protozoa. So I have given in-depth comparison because this is a very important from exam point of view. And questions are more often asked in net examination, mains examination, or two numbers, high numbers, and 10 numbers. One more thing. So there are different sub-organisms. You have to keep in mind their functions. For example, what is capsule? What are its functions in relation to bacterial cell? What is cell wall? Its function, both in prokaryotic and eukaryotic cell. But in case of microbiology, as microbiology is mostly we are dealing with prokaryotic cell. So questions are often asked on the different functions and structural organization of cell wall, cell membrane, uh, glycogen granules, flagellum, pili. So questions are asked on the find numbers or two numbers, means what are the different structures of prokaryotic cell and explain its function. So like this, I have given here. Just this is in brief actually, but you have to study in depth because cell will have multiple functions. Cell membrane have multiple functions, many functions. So you have to keep in mind all these functions. And questions are asked for final assessment. Means I mean, if you are studying very minutely, then if questions come for two numbers, five numbers, or 10 numbers, you can easily write it. So that's why I will suggest you should read very minutely because question nobody knows question may come or may not come, but it will give you confidence that we have that you people have covered in-depth syllabus. So you will be ready to face any type of difficult questions on this different area. So, in prokaryotic, I will discuss about bacteria. Bacteria is one of the major group, and I want to say in ARS microbiology discipline as a most of the questions are focused on the bacteria because bacteria is the core group of ARS microbiology, agricultural microbiology. Uh, Questions are 90% of the questions are focused on bacteria and antibacteria. Uh, Regarding other group like Anja, Protozoa, algae, questions may come, but they are not too much important. So you have to keep in mind. But you have to read all the syllabus. You have to cover all the syllabus. But focus will be on this major area. So this is general features of bacteria, their habitat. As I have discussed, if you know the detailed attributes of prokaryotic cell, then you will easily know the characteristics of a bacteria or archaebacteria or protozoa or algae. That's why you have to keep in mind the detailed difference between these two cells. Because if any questions come, you can write this property, and these properties are applicable to the if they will ask write down the characteristics of archaebacteria, then you can write this property because their properties are applicable to archaebacteria and probacteria and bacteria. If questions are asked on probiotic cell, if they are asking question on algae, then you can write the properties for eukaryotic cell because these groups are included in eukaryotic cell or prokaryotic cell. So these are some characteristics I have shown here. General features of bacteria. They are present everywhere. They can grow with oxygen or without oxygen. Their relationship may be parasitic, symbiotic, or saprophytic. Archaebacteria is a group of bacteria which generally grow in extreme condition. Extreme condition means maybe at extreme high temperature, high salt concentration, high atmospheric pressure. They need to be seen through microscopic observation. Smallest and most primitive organism. They have wide range of shapes. The cell wall is, as I told earlier, cell wall is amino acids and saccharides with some that peptidoglycan is major cell wall compounds in bacteria. One more difference is nucleus, well-organized nucleus is absent in bacteria, in prokaryotic cell, while eukaryotic cell have well-organized nucleus. This is a major difference actually in prokaryotic and eukaryotic cell. Like other organelles which are present in eukaryotic, they are absent here, like mitochondria, Golgi bodies, endoplasmic reticulum. In case of mitochondria, the functions of mitochondria in prokaryotic cell in bacteria is performed by mesosomes. So besides this, there are a lot of characteristics I have not shown here, but you have to read this characteristics because questions are more commonly asked on this area, and you have to keep in this mind. If you keep in mind this figure now, then you will automatically able to write the characteristics. So another aspect is what are the different methods that are used for the identification of bacteria. Here just I have a given list. Some microscopic and morphology, gram staining. Using gram staining, we can divide bacteria into gram positive and gram negative bacteria. Shape there are various shapes like rod shape, local shape, spherical shape, spiral shape bacteria. Arrangement means arrangement of flagella. Flagella on the list of flagella arrangement, there are various classification on the list of motility, microscopic morphology, third point, physiological and biochemical properties. This is the major classification, and questions are asked on this area. What are the physiological and biochemical and nutritional classification of bacteria? So that's why I have discussed here in the slide. I have given the important classification of bacteria based on this aspect. Other approach can be chemical analysis, phase typing. Phase typing means which bacteria, which virus affect the bacteria. Serological analysis can be used for classification and identification of bacteria. Pathogenicity. In genetic and molecular analysis, percentage of GC composition, DNA analysis using genetic probe, and nucleic acid sequencing and rRNA analysis. So these are some basic traits through which particular bacteria are identified and classified into respective groups. So we have to keep in mind there's different classification because this is another important area. Questions are asked on this area. For example, gram staining. People questions can come. What is gram staining? Write down the detailed procedure for gram staining. For most of the time, questions have came for 5 numbers and 10 numbers. So you have to keep in mind keeping this mind, you have to prepare in that way. So here, as I discussed, here I will show you the physiological classification of bacteria based on the some important parameters. So here is a classification of bacteria based on the oxygen requirement. So based on the oxygen need, bacteria can be grouped into 5 groups like obligate aerobes, facultative aerobes, aerotolerant anaerobes, obligate anaerobes, and microaerobes. So just by looking into this test, you can just that obligate aerobies they are growing in high oxygen concentration. Facultative aerobies means they can use oxygen but can survive without oxygen because they are growing at the bottom of the test tube in liquid medium. Aerotolerant anaerobes are those kind of bacteria, those kind of microbes which don't use oxygen, but they can grow in their presence. Obligate anaerobics, as by looking into this test tube, we can say those bacteria which are not able to grow in presence of oxygen because oxygen is present at the surface of the liquid. So at the bottom level, there is a lack of oxygen. So those bacteria which grow in absence of oxygen, they are obligate anaerobic, and that is microbial. Like they they grow in oxygen, but very low concentration of oxygen. Means they use very low concentration of oxygen for their growth. So in the backside, we have to know why particular bacteria grow in presence of oxygen, why particular bacteria didn't grow in presence of oxygen. So their mechanism, you have to know the mechanism. For example, presence of cytochrome deoxygenase enzymes in respiration is required for the bacteria who grow the oxygen. It means to grow in presence of oxygen. So in case of obligate aerobes, these bacteria have presence of cytochrome C oxidase enzymes. So other bacteria they are lacking that enzymes. This basic mechanism why particular bacteria are growing means using oxygen or not using oxygen. We have to keep in mind their basic mechanism because in-depth questions may come write down the mechanism of bacteria growing under oxygen condition or unoxygen condition. So we have to know the actual cellular mechanism of particular bacteria growing under particular condition. And regarding overall classification, these questions may come for 5 numbers or 10 numbers. Likewise, this is a classification of bacteria based on the temperature. Actually, here I have shown different types of bacterial group and their temperature range, like psychrophile, mesophile, thermophiles, and hypothermophiles. Uh, with as a net hypothermophiles mean they are growing at more than 80 degrees Celsius. Thermophiles are those bacteria they are heat stable and grow between 50 to 80 degrees Celsius. Mesophile means those bacteria or microorganisms they are grow under optimal temperature range, that is 10 to 40 or 25 to 40. Psychrophile means uh, bacteria growing 10 to 20, 20 degrees Celsius. So this is a classification you have to define. This is a bacterial classification on the salinity requirement. Means how different bacteria grow in different salinity salt concentration. So here I have given different salt concentration and the name of particular bacteria growing on different concentration. Here again, you have to know the mechanism. For example, question may come. What is the mechanism of adaptation of extreme halophiles under high salt concentration? So here question will be somewhat in more depth. So we have to know the actual mechanism of this bacteria under high salt concentration. So in addition to knowing the this classification, we have to know if you want to get succeed in ERS examination, if you want to have more number of marks in mains examination, this little information, little minute and in-depth information you should know. You should know that, for example, here I discussed now, what is the mechanism of adaptation of halophiles to the salt concentration. So this information you will get in a reference book, like for example, Brock Biology of Microorganism. That is particular book actually. I will recommend that book that every microbiologist should have, or other discipline people should also have that book, because that book have very well covered the basic principle and mechanisms of different microbes that are growing under diversified condition and extreme condition. So you should read that particular book. That is Brock Biology of Microbiology. You should have that book. Likewise, and other thing, and every group you should know the examples of microbacteria that are, for example, you should know the thermophiles, list of thermophile bacteria. Because if question will ask what is the thermophiles, give the examples of thermophiles. So for every group and subgroups, we have to know the examples of microbes which are grouped into particular group. So you have to keep in mind the examples of bacteria or microbes according to classification and their subgroup. Based on the pH, bacteria are acidophile, neutrophile, alkophile. That is acidophiles grow in acidic condition, neutrophile grow in neutral condition, and alkaliphiles mostly grow in alkali condition. So this is a in case of neutrophiles, most of the organisms are hemopathogens, because in case of alkali files, we have to keep in mind the examples. So as I discussed, you should know the representative examples of microbes under a particular group, because questions are mostly asked about two numbers, defines acidophile, neutrophiles, and give the representative lists or examples of microbes in that range. So you have to keep in this mind. Is it going okay, all of you? Hello, hello, all of you listening?
Yes, sir.
Is it going okay for all of you?
Yes, sir.
Okay, okay. I will continue then. This is another classification of macro based on the nutritional requirement. This is a very important classification, because all other microbes, all the macro classification is, if you remember this chart now, then you can easily identify the nutritional types of particular groups of microbes. So based on the nutritional requirement, microbes can be divided into four types. That is photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. So their classification is mainly based on the types of energy, electron, and carbon source. And here I have listed the organism. If you see energy source can be light and chemical. So when there is a when particular organism is using light as energy source, then it is a phototropic organism. The particular organism is using chemical as light energy source. That particular organism is chemotrop. So again, in phototrop, carbon source will be CO2, carbon source will be organism. Here I have mentioned that. For example, here light energy carbon source is CO2. In case of photoheterotrope, carbon source is organic carbon source. In case of chemoautotrope, inorganic chemical compound act as energy source, and inorganic electron donor is cow because it is autotrope. That's why CO2 is using as inorganic carbon source. In case of chemoheterotrope, means organic compounds act as energy as well as carbon, energy, electron as well as carbon source. And their respective examples are, for example, photoautotrophs means all the plant kingdom or uh, algae, cyanobacteria, they are grouped into photoautotrophs. In case of photoautotrophs, purple and green sulfur and non-sulfur bacteria. In case of chemoautotrophs, means they are using inorganic chemical compound as energy source. So organism in this group are nitrogen bacteria or cyanobacteria. And chemoautoheterotroph means mostly pathogenic bacteria, fungi, and pathogen. So we have to do this classification. This is major area, and questions are most of the times questions are asked here. What are the photoautotrophs, their characteristics, and their classification? So you have to keep in mind. Questions are mostly asked for kind numbers and 10 numbers. The question may come for two numbers and objective question. So this is a very important area. Start from objective type question to the 10 numbers questions may come from this area. So you have to keep in mind this classification in detail. Along with classification, their detailed characteristics and this sub example group you have to keep in mind. So you have to prepare in that way. So in case of then I will discuss about prokaryotic group, important prokaryotic group in this prokaryotic bacteria. That is photosynthetic bacteria. Here I have given some examples of photosynthetic bacteria like Proteobacteria, that's purple bacteria, Helibacteria, Chlorothexi, that is flammable bacteria, also known as green non-sulfur bacteria, Chlorobi, that is green sulfur bacteria, and cyanobacteria. So I have not given here care characteristics. So here, but I have given what type of questions are asked on this area. So question as I discussed here, important features of cellulose. Maybe they will ask you what is purple bacteria and they describe their characteristics. Okay. Since this is the bacteria, they are as per we know, photosynthesis is carried out by plants. So, but in case bacteria are carrying out photosynthesis, so they will ask what are the unique or different mechanism of photosynthesis in bacteria and compare it with photosynthesis mechanism in plant. So we have to keep in mind because there are some unique characteristics of photosynthesis in bacteria. So we have to keep in mind that features and we have to prepare by comparison this characteristic with plant like types of how CO2 assimilations occurs. That is type of phosphorylation. I discussed the comparison of photosynthesis versus plant versus algae. We have to keep in this mind actually. Again, cyanobacteria is very important group from agriculture point because cyanobacteria is used as a biofertilizer. So question can come from cyanobacteria. Its historical development means various researcher we have carried out work in the area of cyanobacteria. And what is the role of cyanobacteria? Cyanobacteria we can call it as blue green algae. Both name are same. Cyanobacteria blue green algae. So role of cyanobacteria as biofertilizers. And what are the examples of cyanobacteria? which are used for biofertilizers. This question will come for two numbers. Another thing is mechanism of nitrogen fixation in cyanobacteria. Especially microbiologists people are knowing that in cyanobacteria there is a specialized structure that we can say that is heterocyst. Heterocyst is the site of nitrogen fixation. So questions have came for 10 numbers for my time. Questions have came for 10 numbers. What is a cyanobacteria? What are the sites of nitrogen fixation in cyanobacteria? Describe the structure and functions of nitrogen fixing site in cyanobacteria. So this is a major area where questions are coming regularly for 5 number and 10 numbers, because other regarding other photosynthetic bacteria, they may not be as much important as cyanobacteria, but you have to prepare all the subgroup, but you can have more focus on cyanobacteria, because this cyanobacteria, or you can say BGA, this is a usually as fertilizer, biofertilizer in paddy cultivation. So that's why keep the you have to prepare the keep in mind the mechanism of nitrogen fixation their side, as I discussed. Another thing is role of GS Bogart pathway. This is actually somewhat physiological aspect. Once the nitrogen fixation occurs in cyanobacteria, that ammonia need to be assimilated inside the cell. So this is a GS Bogart means glutamate synthesis pathway. So this is a unique pathway of ammonia assimilation in cyanobacteria. So questions are coming on this area. What is GS Bogart pathway, and describe this pathway and their role in nitrogen or ammonia assimilation in cyanobacteria. Other questions like maybe what are the production technology for BGA based biofertilizers and BGA also as a biofertilizers. So these are some possible questions that are coming in ARS examination. It may in ARS prelim, ARS net, or ARS mains examination. So you have to keep this in mind and prepare in that way. Other group is this is also very important group, chemo photograph. We can say chemotroph. This chemotroph means this is organism that obtain energy from chemical reaction, but the source of carbon is, as you article is CO2. So the source of carbon is from carbon dioxide, and their major groups are like methanogens, sulfur oxidizer, reducer, nitrifier, NMX bacteria, thermoacidophile, iron oxidation bacteria, etc. So, as name suggests, for example, sulfur oxidizer. So you have to keep the role of sulfur bacteria in sulfur cycle. In sulfur oxide, there is a sulfur oxidation and reduction cycle in soil. So we have to keep in. We have to know the role of actually sulfur bacteria in sulfur cycle. Likewise, nitrogen means you have to know the role of means you have to know in detail nitrogen cycles and how different microbes are responsible for the carrying out different steps of nitrogen cycle in soil as well as environment. Like in case of iron oxidation bacteria, you have to know the detail the iron cycle. If you are knowing the detailed iron cycle, then you can easily summarize or you can easily remember the exact roles of particular bacteria or microbe in respective cycles. So question on this area like definitions, important feature of subgroup maybe methanogens, sulfur oxidizers, reducers, chematography versus photography comparison. This question can come. What is the difference between chematographs and photographs? Question how came actually. Mechanism of carbon and nitrogen assimilation in this group. As I discussed, the role of this bacteria in biogeochemical cycles such as sulfur cycle, nitrogen cycle, phosphorus cycles, iron cycle, manganese cycle is also there. What is nitrification, denitrification? Another aspect is environmental and agroecological significance of chematograph. So this is out of box actually, out of box possible questions I have mentioned here. When you are reading particular group of macro, you have to read in that way that you should know the basic characteristics as well as their habitat as well as their agriculture as well as ecological applications. You have to read in 360 angle. Only then you can easily remember and capture the particular groups or overall microbiome, because Microbes are such things we can't see. So by just visualizing and imaging and that correlating their basic property, their application or their ecological habitat, you can grasp more information on this group. So you have to prepare in that way. Actually, this is actually quite from previous slides. This is nitrogen fixing bacteria. I have what he mentioned here characteristics. You have in this nitrogen fixing bacteria, you have to remember the different classification, different types of nitrogen fixing bacteria. That is free-living, anaerobic, macrobically. Uh, Here again, nitrogen fixation is due to the presence of nitrogenous enzymes in this bacteria. So you have to know the what is the nitrogenous enzyme complex, their mechanism of nitrogen fixation in bacteria. Okay, that's why you have to study in that way because nitrogen for fixation of nitrogen in root nodule bacteria, they have nitrogen enzyme complex. So we have to study the mechanism of nitrogen enzyme complex, and this is a very important area because this is can somebody mute their audio? This is a very important area because this is a core of agriculture microbiology. So you have to prepare in 360 angle. For example, anaerobic nitrogen fixation and aerobic nitrogen fixation. So high particular bacteria fixes nitrogen in the anaerobic condition. You have to know the mechanism of nitrogen fixation under anaerobic condition, under macroaerobic condition, under aerobic condition. That's so that you can prepare in between. Their examples, their symbiotic, you have to know endophytic bacteria, symbiotic bacteria. These are examples. And one more thing is root nodulation. For during this time, questions have come for 10 numbers. Discuss the important steps in root nodulation and mechanism of nitrogenous enzyme complex in rhizobium for fixation of nitrogen. So this is a very core group. I think some other people from soil microbiology might have taught this aspect during this training. So I'm not going to detail, but this is very important point. You have to keep this classification in classification, their mechanism, their host name, host plant name, and other related information. This is actin acids, some other actin acids actually. This is actually in between fungi and bacteria. So these characters are actually it is the structure is prokaryotic, but morphology is filamentous type. So it is intermediate between bacteria and fungi. It is a gram-positive bacteria. Respiration mostly aerobic, some can go anaerobic. Their habitat is soil and marine. Why I have given here function because this is a very important group of microbes actually. Questions most can come for what is the application of active acids. So this application means their functions. So their functions can be in soil condition. They are source of antibiotics because most of the antibiotics produced and extracted from antibiotic active acids group like after rainfall we can say we can have smell earthy smell of earth, so that smell is due to the geosmin compound. So questions more often comes what is the name of compound which is responsible for uh, giving earthy smell earthy water to the soil and water. So this type of questions we have to know. Like other functions, they are mainly involved in the degradation of organic molecules. They are capable of some species like Frankia. They can fix atmospheric nitrogen. Uh, Here I have given this picture. I have given actinomaces and their different product. They have some medicinal use, environmental use, or industrial use. So you can read these slides, and you can also read from other references. This is actually a picture you might have seen. This is actually somewhat mycelial picture. Uh, because in case of bacteria, the colony growth is like slimy in nature. But this is active acids as it is a their properties are intermediate between bacteria and fungi. So their mycelium means their growth is like liquid mycelium, filamentous type of growth is there. Again, we have to know, as we know, actin acid is prokaryotic. But actually, we have to keep in mind the differences between bacteria and actin acids. Questions often comes: what is means what is actin acids, their characteristics, and differentiate it between actin acids and bacteria. So we have to know the difference between actin acids and bacteria. Here I have given the differences like actin acids is filamentous, while bacteria is not filamentous. Their order is different. Gram positive bacteria may be gram positive or gram negative. Faculty anaerobic bacteria are bacteria have all group like aerobes, anaerobes, or facultative anaerobes. Shape is oval shape. Bacteria have all kind of shape. Like growth characteristics, they form powdery colonies on that stick thermal plasma. As I discussed, bacterial colonies are slimy and distinct. Their growth is slow as compared to the bacteria. And so bacteria growth is very fast. Again, one more thing is that is the form hyphae and conidae as similar to the fungi. So this property is similar to the fungi, but it is a different from bacteria. And one more thing is one more major difference is actin-acids are non-motile while bacteria are motile. So there's differences and their points you should know. Uh, these questions can come. Means if you know the difference, you will know the characteristics of particular bacteria or actin-acids as well as it will be easy for you to compare actin-acids with bacteria and fungi. So this is also important table actually. It compares with how bacteria, actin-acids, and fungi are similar and differentiate with each other. So I'm not going to teach this. Now another important group is spore-forming bacteria actually. This is also a very important group actually because this group is responsible. They are able to produce resistance spore, that is endospore. In during our previous during our initial slide, as I told you, that John Tyndall was responsible for endospore, development of endospore or discovery of endospore. So that endospore is mostly formed by bacteria. So this in this case, I have shown here a picture the steps in formation of endospore in bacteria cell. So this is important. Questions came for kind numbers. What are the endospore and describe the steps involved in the formation of endospore in the bacteria. So endospore is actually a type of resistant spore which are I told you which are resistant to alkali condition, acidic condition, and temperature, uh, and other extreme parameters, other extreme factors. So you have to keep in mind the different steps that are involved in the formation of endospore in bacterial cell as well as at the same time we have to know the features of spore forming bacteria, their list examples of list, and same time you have to know the how particular spore forming bacteria are different from non-spore forming bacteria. So that's why I have given here differentiation comparison between spore forming and non-spore forming bacteria. So you have to know the this differentiation. Another important group is lactic acid bacteria. This is a very important group in food and industrial microbiology. So this is a you have to in this bacteria you have to know their characteristics. That is probiotic means as you know in the pickling or yogurt or curd, the major role is lactic acid bacteria. So there they have diverse part application. Their uses you have to know the how lactic acid bacteria is used in different area. Their probiotic characteristics. Probiotic means they are beneficial to human. And they can improve our gut microbiome. So what are the unique characteristics or what are the beneficial characteristics of the bacteria which are used as a probiotic bacteria? So you have to know the different features and their characteristics. Questions come for 5 number 10 numbers. So in this regard, you have to put their characteristics their applications area. Like they can have application area maybe food and feed industry, chemistry, uh, lactic acid bacteria, source of different functional ingredient like enzymes, vitamin, exopolysaccharides, low calorie sweeteners. It can also act as antimicrobial agent like biopreservative, medicine sector. So like this, you have to read because this is a also important topic. The questions come from questions may come from high number of different. Again, actually, in lactose bacteria, they have two major roles: homofermentive and heterofermentive. Homofermentive means they produce similar type of compounds, and in case of heterofermentive means during fermentation process, they can produce different. Now, as name suggests, hetero means different, two types more than two. Homa means similar. So this is the also very important comparison. We have to keep in mind what are the lactic acid bacteria. Their important groups are homofermentive bacteria, homofermentive lactic acid bacteria, and heterofermentic lactic acid bacteria. So here I have shown uh, the difference between homofermentive bacteria and heterofermentive bacteria. So we have to homofermenting is as name suggests, they only produce glucose, monosaccharides. It may be glucose or fructose. In case of heterofermentive, they may produce glucose, galactose, and other sugar. So that alpha and beta links are present in heteropolysaccharides. Uh, they are also present in typically branched linkages are there. In case of homopolysaccharides, uh, linear may be branching may be linear or branched. Their example, you have to keep in mind the examples. Question may come, what are the hemoformative and heteroformative and describe their characteristics, the suitable example. So you have to keep in mind suitable examples of homo and heteroformative. So I'm not going to read this all characteristics. I will, as I told, I will give this presentation to you. You can read this, or you can read from any book or internet source. In prokaryotic cell, uh, till now I discussed about bacteria and different bacterial group, their classification. In prokaryotic cell, second best group is archaebacteria. This archaebacteria is similar. They have more similar to bacteria, and but their major feature is that these archaebacterial organisms are present in extreme conditions. These organisms are present in extreme conditions means extreme pH, extreme acidity, it means extreme growth conditions. So their cellular organelles or their cellular structures are in such a way that they have adaptation to grow under extreme natural conditions. So you have to know the what are the cellular adaptations of this archaebacteria that have made possible them to adapt under such high extreme conditions. So here I have given their characteristics. They are like the cell membrane have lipid. As lipid is more resistant to extreme condition, so lipid is major component of the cell membrane. Uh, another major aspect is that their cell wall is made of pseudomembrane. Actually, in case of bacteria, their cell wall contain peptidoglycan. Peptidoglycan compound as one of the major component of cell wall. While in case of archaebacteria, in order to grow and sustain in extreme condition, extreme natural condition, uh, this bacterial cell wall have pseudomembrane. And this is responsible for this compound protect cell and it avoid the burst the damage to the cell, damage to the bacterial cell under high extreme condition. So you have to keep in this mind the typical features of this archaebacteria. Like bacteria, this archaea don't have membrane-bound organisms like nuclei, endoplasm, reticulum, mitochondria, chloroplast, etc. As I told you, habitat extreme environment or extremophiles, uh, they can withstand very high pressure of more than 2 atmospheric pressure. So this is due to the cellular adaptation. So what are the cellular adaptation? We have to know what are the actual cellular adaptation that are making Archaebacteria responsible for growth under this extreme condition. One more unique feature is that this group of bacteria is indifferent towards major antibiotic due to the presence of plasmid which have antibiotic resistant genes. So this is actually figure. Other characters like as I told you, they lack like ribosomes are more similar to eukaryotic. Means ribosomes are of 80S type rather than 70S type, which are present in prokaryotic cell. Plasma membrane as well is made of lipid, which are different than most other organisms. As these archaebacteria have to sustain under extreme good condition, their cell will have some unique component which are different from other organisms. Regarding the questions, question may come, what are the archaebacteria, their characteristics, uh, different comparison between archaebacteria and bacterial characteristics. What are the different subgroup of the archaebacteria? Their subgroups are thermophiles, acidophiles, alophiles. Again, in subgroup, question may come from what are the methanogens, their characteristics, their ecological significance. For example, methanogens during the biogas production, uh, conversion of organic material, which are subsequently converted into biogas. In that biogas preparation, mostly during later stage of biogas, there is a more of anaerobic condition. So under anaerobic condition, this bacteria play important role in conversion of formed CO2 and alcohol into methane. So in this perspective, you have to correlate their significance and their application area. Like thermoacidophiles, like we have to know their characteristics, their industrial importance because in case of thermoacidophiles means these archaea which mostly grow at higher temperature and higher acidic condition. Temperature means they can grow at almost 100 degrees Celsius or 120 degrees Celsius. So their applications or importance may be many of the industrial enzymes which require high temperature or which have to be used in high temperature. So these types of enzymes are isolated from such types of bacteria. So these types of application we have to keep in mind. We have to remember because questions may come. What are the industrial importance of thermoacidophiles? So we have to be prepared in that. Here again, I told you this archaebacteria, it have some similarity to bacteria. It have some similarity to eukaryotic. Eukaryotic is fungi. So you have to know the similarity between bacteria and archaebacteria. At the same time, comparison or differentiation between bacteria and archaebacteria. So here I have given the tables. Uh, for example, the similarity between bacteria and archaebacteria is the absence of nucleus and micro-organisms. The characteristics which are typical for archaea and eukaryotics, but different from bacteria, means absence of peptidoglycan. In case of character which are only for archaebacteria, means uh, the cell wall of this archaebacteria contains pseudomerine. This pseudomerine is only present in archaebacteria, while it is absent in bacteria and eukaryotic cell. So in that way, you have to know what is archaebacteria. It's a similarity with fungi, that is eukaryotic, as well as bacteria. At the same time, it's a differential character from bacteria and eukaryotic. So I'm not going to read. I'm not going to read in details the comparison. So you have to keep in this mind. Here again, the differentiation between bacteria, archaea, and eukarya at the cellular level. Like in case of chromosome, bacteria and archaea have separate chromosome. In case of eukarya, it is a linear chromosome. Uh, in case of chromosome segregation, or we can say reproduction, is binary fission. In case of eukarya, it is a due to mitosis. Ribosome type bacteria archaea have synthesis ribosome. In case of eukaryotic ATS. So, in case of translation process, initial tRNA is formal methionine, while in case of archaea and eukarya, it is a methionine. So, these are some difference point you have to keep in mind. The difference between bacteria and eukarya, as well as similarity between bacteria and eukarya. So this was about bacteria. One more microbial group is mycoplasma. This is also important as it have given in our syllabus. So questions often come for high numbers. What is the mycoplasma? Again here, you have to keep in mind the characteristics of mycoplasma. Their comparison, with bacteria. Their mostly as this bacteria, this organism have different characteristics, some unique characteristics. So most of the times questions came regarding the mycoplasma, their characteristics and the types of disease caused by mycoplasma. So one of the and their discovery, who discovered this? So in 1898, Knockard and E.R. Ross, they discovered this kind of bacteria. But this is actually bacteria, but that lack cell wall. So due to lacking cell wall, it is pleomorphic, means it can take more number of shapes. So these are some characteristics that is like smallest cleaning organism, lack cell wall. Their size is uh, low. Danish naked ribosomes of cementase type. One of the most unique characteristics is pleomorphic, as due to lack of cell wall, they can have different cells. Other important thing is that they can cause plant disease actually. Plant disease like bunch top of papaya, citrus greening, little leaf of brinjal. So questions have come. Write the examples of disease caused by mycoplasma. So in this regard, you have to keep in mind all the information regarding this mycoplasma, their discovery, their typical characteristics, and disease caused by them. At the same time, you should know the trigger, because if you have to score for any discipline, it is very important, because if you have to score extra numbers in mass examination, try to have figure, image, or any chart type of information, because in that information you can cover more information within short time. So it is better to include any figure, graph, diagram. So this is the one of the suggestion you can have. We can take from this lecture. Another thing is fungi. Actually, fungi. This is one of the major group of eukaryotic. I will not discuss this fungi. This is eukaryotic cell, and actually this fungi question may come from hand numbers. So we have to read this their properties. As I will discuss in the previous slide, that is eukaryotic cell. So whatever the properties eukaryotic cell have, that are applicable to fungi, protozoa, or algae. So you can keep that information in mind. Along with some individual groups of fungi have some unique characteristics. So you can keep in that mind. So this is a fungi information regarding fungi. This is algae. Algae may be unicellular or multicellular, photosynthetic. They are grouped on the basis of pigmentation, storage products, or composition of cell. Like protozoa is single-celled eukaryotic, similar to their nutrition need is similar to animal. They mostly freely live in water, animal host. Their reproduction may be asexual or sexual means. Locomotion is by due to pseudopodia, cilia. So you have to read this. You have to read this all basic properties. Virus is important for various discipline in microbiology. Virus is very important, and questions often come for high number 10 numbers. What are the virus, their characteristics, and do virus are living entities or non-living entities? So while giving this information, you have to compare why virus is living. You have to describe why virus is non-living. Why virus is living? Because if you have a nucleic acid may be either DNA or RNA. It have protein code. It can replicate inside the living cell. So this all property is of living organism. But why living non-living? It is non-cellular, because it don't have typical cell. It cannot reproduce themselves. Don't have any cell organelles. Have only one type of nucleic acid. It may be DNA or RNA. Don't feed on the waste time. So this difference you have to know. Questions often come here. Their characteristics I have here mentioned, like virus have inner core of nucleic acid, maybe either DNA or RNA. They have protein code. Their size range is 20 to 50 nanometer. They are inert nuclear protein, filtrable agent, obligate parasites. They have crystalline crystallinity, and they can multiply only inside the living cell. Means they are not capable of multiplying outside the living cell. Metabolically inefficient means they have to depend on other host cell machinery for their reproduction. So this is a figure actually. This is the nucleus, viral genome, nucleocapsid, innular protein. So along with property, you have to keep in mind the figure of particular organism. If you are giving particular figure now, then it gives catchy for the examiner. So you can automatically fetch more number of marks than your other competitor. So you have to include figures in every whatever possible. You try to include figures, such kind of figures in your question answer sheet. So this is the lytic and lysine cycle, how virus multiply. In lytic cycle, virus generally when grow inside the host cell, we generally have multiply, produce large number of copies, and it get released from host cell by bursting their cell. So in case of lytic cycle, virus actually virus genome get integrated into host genome, as I have shown here, and they remain with host cell in silent mode. So these are figure their difference. So you have to know the difference between you have to know the definition of lytic cycle and elastic cycle, their differentiation. There is some characteristics. So this is also important area. Questions come for high numbers. Difference between lytic and elastic cycle. So you have to keep in mind the figure as well as various differentiation point between lytic and elastic cycle. So these are some questions which can be asked regarding virus, the general features of virus, how different from bacteria, virus structure and morphology, how virus is cultured, and techniques used to identify viruses. Virus multiplication cycle, maybe lytic and elastic cycle. What I have now discussed here, specialized transduction, DNA containing virus, latent viral infection, persistent viral infection, and primes. Primes I will discuss here. So this type of questions may come. We have to do iron. Iron is actually RNA, but it don't have say what. So this is a it also contain either RNA or DNA. So this is actually characteristics of virus. We have to read this virus too, and uh, you have to know the difference between virus and virus. Because question have come, what are the properties of virus and virus, and differentiate between virus and virus. So you have to know the difference between virus and virus. Here I have given virus and virus. That I discussed here. This virus is nuclear protein particle, means it have nucleus plus protein core. Virus have only RNA particles. You don't have any protein. Small particle, virus is smaller than virus. Virus contain either DNA or RNA at their genetic material. But in case of virus, it contains short strand of circular single stranded RNA as genetic material. Again, as protein code, virus or protein code, virus don't have protein code. Different type of virus can infect different hosts like animal, plant, or bacteria. But the typical feature of virus is it only infect plant cell. Uh, Other feature is this virus particle produces protein during their replication, and this virus particle don't produce. Example, we have to give example also. In case of virus, the typical example is potatoes, internet, people virus, and other related virus. So we have to keep characteristic their differentiation versus virus, as well as the disease caused by the virus. Price are in our section. Price is last group of matter. So this is not included in syllabus, but questions had came during our time. What is price? So price is actually this price is organism. It don't have any nucleic acids. Means nucleic acid is absent actually, and it have only proteins. So this is also called as infectious protein. So prime is also called as infectious protein. So you have to know the characters of primes also. Question in this area, questions mostly for objective or two number question. 5 numbers and 10 numbers questions are not expected on this area. So I will suggest you to have to read the characteristics of in detail characteristics of primes also. So this was all about my presentation. In last presentation, I will suggest for ARS microbiology, I will suggest to have this reference book for reading, because most of the questions, not most, almost all the questions are coming from these books. For basic microbiology, the books of general microbiology by Roger Steiner. I told you Brock Biology of Microbiology. It is a very book actually. It's medical publication book. Classical Microbiology is another basic excellent book. So these are very excellent book. They have covered very in-depth. They have given very in-depth information. Basic Microbiology by Pelzer. So all these books you can have. Regarding soil microbiology, you can read soil microbiology information from soil microbiology and agriculture microbiology by the books of N. Subbarao. And in addition, some chapters related to the soil microbiology can be read from the basic microbiology book, because these books have covered up all the aspects: basic, uh, basic microbiology, food microbiology, industrial microbiology. So some aspect can be read from this microbiology, some basic book actually. In case of food and industrial microbiology, you can have book like food microbiology by Frazier, food microbiology by Adams and Moss, and industrial microbiology by Cassidy. So these are reference book. I suggest all the candidates who are preparing for ARS in microbiology, because we people also have used these books for our ARS preparation. So because these books are very sufficient and questions only come from these books. So I will suggest these books for your references. So these were my all for my today's presentation. In addition, if you have any queries or any if you want any guidance, you can contact me on my this address. I have my email here. Thank you all. If you have any questions, any query, you can ask me. Hello? Are you listening to me?
Yes, sir.
If you have any questions, query regarding exam or the relevant information, you can contact me or you can now ask me.